Basophils. These were not in high number in the white blood cell count. However, if we need more, the body can produce more. The basophils, their secretions, their superpowers that they have, they act as a vasodilator. Once again, breaches that occur. Breaches are going to breach capillary beds. Capillary beds are the area of exchange, pretty much where everything takes place, okay? So, if there is vasodilation, which does occur at the time of a breach, what does that mean? Have the capillaries opened or closed?
Closed.
They have opened.
Open.
So, if they have dilated, this means I'm getting more blood to the area. However, the chemicals that have been given off are saying I need these cells. So it calls in these cells of defense as well as all of the stuff that's in the blood to help heal the area. Does that make sense? So the basophils, they're going to act as the vasodilator, getting those capillary, those vessels to open so that we can rush more material to the region. This aids in getting more leukocytes to the area. In some areas of connective tissue, we find those mast cells. Do you guys remember those? Like along the blood vessels, for example. Their job is to do the same thing. Make sure that we dilate the blood vessels and rush blood to the area. Lymphocytes. They're going to become so interesting for the lymphocytes. When you see them, they're going to look similar. So people who do this all day look at blood, and they can view all the cells that are in the blood. They're going to know how to categorize those lymphocytes. The lymphocytes are going to get categorized into natural killer cells, T cells, B cells. Natural killer cells, pretty cool, okay? But the point, because those are very active in the general defense system. We're going to see these get a little more interesting once we get into specificity. Okay, this is going to be cells that we need, and they create immunity. The monocytes, those are lymphocytes that emigrate from the blood into the connective tissue. So when it moves, it transforms into a macrophage. Some are wandering around. Some of these are going to be fixed. They can be fixed in the tissue, and they're especially fixed in the organs. For example, the lymph nodes. Now, I put the line in here because basically this moves to the next topic that was in your chapter, all right? After it talked about these particular cells, because one of the things that we're looking at, okay, we're going to have to go from non-specific to specific resistance, but something else that's important in non-specific, antimicrobial proteins. We're going to find that in our body, we're going to have multiple types of proteins. Their job is to help us inhibit the reproduction of these little microbes, these little foreign pathogens that are going to try to invade us. However, it's only a short-term solution. Now, if it can handle the problem, the pathogen, we're good, all right? If it doesn't, might need a little more help. So, it is non-specific, meaning if it encounters a bacteria or a virus, it's going to act the same way every time. There's no specificity, no memory. It's going to attack it the same way every time. Doesn't matter if it's a bacteria or a virus. The two types that we have, interferons and then something called the complement system. Ever heard of either one of those?
I'm assuming they comp.
Has to do with, yes, which one? The complement?
Sorry.
Andy? No? This is going to be, think about what we're looking at. Non-specific. Our response being the same, okay, it's going to include the response of stuff like pain, swelling, redness, fever. And all of these little chemicals play a part in it. And when we have the non-specific resistance, well, the thing about it is the body just wants to get rid of it. That's it. And the hope is that my general defenses can do that. Now, interferons. Notice any particular term right there?
Interfere.
All right, they're going to interfere.
They're responsible for fever, right?
Okay, but what is it acting against specifically?
Iris.
A virus. Are a virus and bacteria the same thing?
No.
No, they are not. What's the difference?
Virus you can't, virus you can't cure or give medication to get rid of. Right?
For the most part, medications are not effective. Maybe I should change that to say antibiotics are not effective against a virus. We do have, for some of the viruses that we can be exposed to, okay, like the herpes virus and shingles. Some of the viruses that we do have, they have created antiviral medications that you can take when there's like an outbreak. Like if you have herpes, you can take this antiviral, and it's more of a boost to the system to help fight the virus. It's still not curing it because you can't, okay? Now, other viruses that we get exposed to, let's say maybe the cold virus, flu virus, maybe like a virus that's just going to be like maybe stomach upset. You know, you have all of these viruses that you're getting exposed to every day, okay? Sometimes you get sick, sometimes you don't. A lot of times we have to depend on the body to mount the attack, okay? Interferons, okay? These are for cells infected with a virus. Now, what we're going to see, they're going to secrete their product. However, when a cell gets infected with a virus, there's a couple of things that can happen, all right? So let's say this is my cell, this is my virus, all right? My cell, now think of this as being one of your body cells, okay? My cell has a nucleus. In the nucleus are my chromosomes, all right? Viruses usually work by one of two methods. They either inject, they'll inject their genetic material into the cell. When they inject their genetic material, one of two things can happen. The genetic material can go into the nucleus, take over chromosomes that would produce more viruses, and every time this cell duplicated, it would duplicate with the chromosome to make more viruses. So if I make more viruses, I can attack more cells. I can make more. Do you see where I'm going with that? Now, sometimes it can inject material so that when the cell bursts, the cell has produced millions upon millions of the little virus molecules, and when the cell finally bursts, these get to go to another cell and start the process all over again. Now, the goal, stop this process. No matter which way it's happening, okay? Stop the process. So the cell that has gotten infected will produce an interferon. This cell is already infected. This cell is going to die in some way, all right? Now, what it's going to do when it produces the interferons, that's alerting neighboring cells that there is now an invader. There is a virus in this area of the tissues, and the interferons that got produced here in the dying cell, it's almost like their last words. They're telling all of their neighboring cells, hey, a virus is here, begin to produce your antiviral proteins. Does that make sense? So, Hopefully, the cells that are surrounding the dying one will produce their antivirals. Hopefully, it will begin to break down the genes, the genetic material in the virus, or stop it from replicating. Do not make more. The interferons that get produced, they'll activate natural killer cells and macrophages. That means these cells will come into the area where this body has been breached, hopefully destroy the cells that have the virus in them. The other thing that's important, interferons are important in our resistance to cancer because activated natural killer cells will destroy cancer cells. It's almost like, well, they studied this because of a virus that caused cancer, and that's how we found it out. The virus actually created cancer in the cells. So we know that the natural killer cells are important in helping to kill cancerous cells.